Joining me today are three University of New Mexico researchers working on a vaccine against the coronavirus. Professor Bryce Chakarian, Vice Chair, Department of Molecular Genetics and Microbiology, Professor David Peabody, Department of Molecular Genetics and Microbiology, and Stephen Bradfute, Assistant Professor, Center for Global Health and Internal Medicine. Thank you all for joining us here on the Zoom. I want to start with you, Stephen. Your lab has a sample of the coronavirus. What kind of work have you been doing so far with that sample that contributes to our understanding of the virus?
We've been doing a lot of testing of different therapeutic drugs to see if these drugs can inhibit viral infection of cells. We're also looking at some basic biology of how the virus interacts with the cells that it infects. And then we've also been using this to test different ways to decontaminate personal protective equipment, see if it inactivates the virus using different surfaces and disinfectants. Those are the types of assays that we've been running.
What kind of work are you doing around a vaccine with the virus?
Right, so we'll be working with Dave and Bryce, and we'll be testing the blood of the vaccinated animals to see if they contain antibodies that can block the ability of the virus to infect cells.
And I want to ask either David or Bryce, there are at least 115 vaccine candidates around the world that researchers are working on right now, and a variety of approaches. I read an interview in the New York Times that the fastest vaccine ever developed was the mumps vaccine, and that took 4 years. Is that the kind of timeline we're looking at?
Bryce, you want to deal with that one?
Sure. So yeah, that's absolutely true that traditionally it's taken a long time to develop vaccines. But I think, I think we're hopeful that developing a vaccine for coronavirus will happen much faster. I think it was very exciting that, just a few months after the virus was actually identified, there was a company from the US that actually was able to put a vaccine into humans. And there have been a few other vaccines that have gone into clinical testing quickly. And, I think that really reflects advances in molecular biology that have allowed for the more rapid development of different vaccine candidates. And, all of these different vaccines have, some pluses and minuses. The ones that we're seeing in people right now are the ones that are easiest to develop. But whether those will also be the vaccines that induce the strongest immune responses, I think that remains to be seen. It also remains to be seen whether we need very, very strong immune responses in order to get protection against coronavirus. So, there's still a lot of questions about, whether these strategies are going to be effective, or if they're going to be perfectly effective, or just be able to reduce the symptoms of disease. And I think either case would be a benefit to people. But it's kind of an exciting time for vaccine development because of this, the intense problem of coronavirus it's really accelerated the progress. And I suspect that we're going to see a much faster timeline than we've seen historically.
I could just add something to that. The advances in molecular biology and immunology have made it dramatically faster to develop vaccine candidates.
Why is that?
Well, because basically the ability to identify an appropriate antigen and to produce it has benefited enormously from technological advances, primarily in molecular biology. And but we still are faced with the relatively long process of clinical trials to ensure that the vaccine is safe and effective. And that really has become the overwhelming limitation in terms of how rapidly a vaccine can be produced.
Now, I know it's called the novel coronavirus because we haven't seen it before, but we have had many coronaviruses, including the SARS. I can't remember what its number was, but I think you worked on that, David. How does our knowledge of those viruses, or any of you, jump in, help us with trying to develop a vaccine for this one?
So the SARS virus from the 2002 or 2003, from that outbreak, that of course was I think, the first really dangerous coronavirus that we have encountered. And because of work done with that coronavirus, we were prepared to some extent for this one, in the sense this one turns out to be quite similar in many respects to that one. And so, for example, the vaccine that we're trying to develop is based on knowledge of the human immune response to that original SARS, and now the one that's called SARS-CoV-2. We're basically targeting similar portions of that virus, as we know were components of the immune response to the original SARS, the one that I think now people are calling SARS-CoV-1 probably.
And Bryce, you have been on the New Mexico Focus before, talking about a vaccine you were developing with your colleagues around Alzheimer's. You're using the same kind of platform technology, I think. Can you talk about how you're adapting that for this kind of research?
Sure. So, the things that my lab works on is using this vaccine platform technology. We use something that's called a virus-like particle, or a VLP. And basically what that is sort of the empty shell of a virus particle. And we work with Dave's lab to use this type of virus called a bacteriophage. So these are viruses that are not infectious to people. They normally infect bacteria. And the reason that we use these virus-like particles for bacteriophage is that we've developed a number of different ways where we can engineer the surface of these particles to display various targets. And so the idea is that we take some antigen, some piece of a virus or another target that we're interested in inducing immune responses against, and then we engineer these virus-like particles so that we can display these pieces of that target on the surface of the virus-like particle. And because these particles have a sort of unique geometry, it turns out that the immune system responds to them very, very strongly. And so we can get these very strong and targeted immune responses against a whole variety of different targets. And so we've done this with a molecule that's involved in the pathogenesis of Alzheimer's disease. And in this study, what we're doing, what Dave has done, is taken small pieces from this novel coronavirus and displayed them on the surface of our virus-like particles. So again, the idea is to use this platform to induce very strong, targeted responses against parts of the coronavirus that are involved in a critical step in its life cycle. So that's the idea behind this project.
In effect, we take a harmless to humans at least, virus-like particle, and decorate it with bits and pieces from the coronavirus, so that in effect it masquerades as the coronavirus to the human immune system, but in a way that's entirely harmless to humans. So the idea is to provoke an immune response to the coronavirus by the display of bits and pieces of coronavirus on this harmless virus-like particle.
Is part of this work figuring out what pieces of the virus generate the best response?
Yes, that's the bulk of what we'll be doing over the next year or so, is trying to decide what are the most efficacious targets, what are the ones that represent the greatest vulnerability on the part of the virus, and exactly what is the best configuration for us to present them in on our platform. So optimizing that is going to be the effort of the next months at least.
And Stephen, where does your work with the actual virus in your lab enter? Where does that come into this process?
Right. So Dave and Bryce had come up with different vaccine candidates, and they'll vaccinate animals, and then we take the blood and try to determine which flavor of the vaccine they come up with is the most potent for inhibiting the ability of this virus to enter cells. And then further on down the line, then we can use mice that are carrying a human ACE2 receptor, which is the receptor that the virus binds to, and then we can infect those mice after they've been vaccinated by Bryce and Dave. We can infect them with the virus and see if it stops the virus from spreading in the animals.
And are you doing this, Stephen, in addition to your regular work? You're doing this on the side.
Right. I think a lot of us are trying to balance that. We do a lot of hantavirus work and some Ebola virus studies and alpha virus work, but wanting to focus as much of their efforts as we can on coronavirus while we can, and then try to balance the rest of the work as well. So I think it's a balancing act for most everybody out there.
I know there are also people at UNM working on genomic sequencing of the virus. Has genomic sequencing also been one of the factors accelerating vaccine production over the last decade or so?
Well, the technology that has led to the ability to rapidly sequence genomes is the reason we know as much as we do about this new virus at all. So the appearance of the genome sequence, which I think was in January, was what enabled all of these sort of new generation vaccine efforts, including ours. In the absence of knowledge of the genome sequence of this virus, we would be completely in the dark.
In the research for our discussion, I read that all the traditional processes of vaccine development are being accelerated. How is the urgent nature of this pandemic kind of changing the way a vaccine might be created?
Well, I think that probably the best example of that is this Moderna vaccine that has entered clinical trial. I think the first patients were in Seattle who were given this vaccine. So this is a new type of vaccine that's based on what's called mRNA. And so mRNA is basically a sort of form of nucleic acid that codes for proteins. And in this case, the mRNA encodes one of the viral proteins. And there's been some very promising data using this strategy in animals, but no mRNA-based vaccines have gone into humans. And so it'll be very interesting and exciting to see how well that vaccine works in people. And I should say that, all the other sort of, classical vaccine techniques are being applied for coronavirus. So there are vaccines that are based on DNA. There are vaccines that use viruses as vectors to introduce the vaccine into people. There are more classical vaccine approaches that use viruses that have been inactivated. I think there's a vaccine trial in China that started using that strategy. And then, of course, our strategy, which is a sort of, another potential approach for targeting what we think might be critical parts of the virus. So like I said, I think it's heartening to see, so many different and diverse efforts to go after this virus. And it could be that all of these approaches may work. And, again, there are going to be issues with vaccine manufacturing once we find a vaccine that works. And so I think having multiple avenues towards a successful vaccine is going to be really helpful in the long run.
You all have a lot on your plate already. There are hundreds of people working on this vaccine around the globe. Why did you feel compelled to also get involved in it?
Well, our platform is different than most others, and so partly I think we saw it as a challenge to our technology, and can we apply our technology to rapidly respond to this? I think part of what we're concerned about is not just this pandemic, but the next one. And the world should be very concerned about the next one. And technologies like ours that have the capacity, we think, to very rapidly respond can in the future play a really important role in heading off the kind of thing we've seen spread across the globe in the last couple of months.
Well, I want to thank you all for taking time out of your research to come and talk about this.
Our pleasure.
Thanks for having us on.
Thank you.